So it is, we're going to change a little bit, still talking about serological surveillance, but changing a little bit in the focus now, not talking about multiple diseases that we can test all at the same time, but what are the results that we can get when we focus in only one disease. And although it's a serological surveillance, it is using some techniques that are a little bit different than Colleen showed, but using the same basis. So what I'm going to talk today is about how we use modeling, especially spatial methods, to understand leptospirosis epidemiology in the Caribbean region. Most of this, go back before, most of this was part of my PhD that I recently finished. So stay with me, we're gonna get through this. So before I start, I'd like to acknowledge the traditional owners of the land and pay my respects to past, present, and emerging. But I also would like to take this moment to extend my respect not only to the traditional owners in Australia, but also the traditional owners in the Caribbean region where this study was conducted. That is the Taino people that were the predominant population when European invasion occurred. So it's a beautiful thing to do here in Australia, but I also acknowledge other lands that were occupied. So today, in the next 15 minutes, I'll be sharing with you some examples of what Colleen showed that we can do with serological surveillance. So we're going to give a very brief background on leptospirosis because I think most of people here would at least know the disease. I'll give enough information to understand the presentation. And then what are the challenges that we found when we were talking about case-based routine surveillance, and why we thought that serological surveys were a strategy to overcome those challenges. And then I'm going to talk about the data sources and the methods that I use and the results that these methods would provide. So, okay, so leptospirosis is the most widespread zoonotic disease in the world. There was a study from Federico Costa that estimated that at least 1 million cases of leptospirosis around the world, with nearly 6,000 related deaths. And in this study, the Caribbean region was identified as the third most affected region in the world, with other studies showing that between 1970 and 2012, Latin America was responsible for a third of all outbreaks of leptospirosis, with the Caribbean region alone representing 16% of all the outbreaks. And other studies showing that 2/3 of all global alerts of leptospirosis came from this region. Those studies showed also that there was some shifts in epidemiological pattern of the disease. So what we knew in the past, and it is still true, but might be changing, is that the disease is mainly associated with occupational risk, mostly in rural settings. So people working with farming, livestock, and others. But this was true until the 90s when it started to decline. And it's also when we started seeing outbreaks being more and more associated with heavy rainfall, followed by floods. We also know that climate change is changing. It's intensifying those extreme weather events, with heavy rainfalls and floods being more and more frequent. And that small developing islands are also more vulnerable to climate change. It means that they have less resources, both economically and environmentally, to overcome the impact of climate change. But we also have less studies and less research occurring in those areas. And we also know that biodiversity loss increase the risk of leptospirosis. Mainly when we lose mammals, we also see the countries being more prone to leptospirosis outbreaks. All of those things combined led us to understand that why the Caribbean region is so prone and so vulnerable for leptospirosis. But at the same time, we can see based on the dates of those studies that we don't know the current outbreak of leptospirosis in the Caribbean region at this moment. And those are images just from the last huge hurricane that affected the area, that was in November last year, showing how it is more of how sensitive we have to be about these, because there's things to be done. And that's what we're trying to do. So the first step, when you think that there is something that's not updated, you're going to search literature and try to find if there is enough things to update that. So the first thing was doing that. So we did a scoping review and complemented with the gray literature to identify all the cases and create a model based on routine-based surveillance to see if we could model this, the incidence of leptospirosis in the region. So we included in our model some variables that are associated with leptospirosis, as you can see here, and that would give us the results of leptospirosis. And it is true, it gave us. So not using surveys now, still using surveillance, I was able to create a model that would predict cases of leptospirosis across time. So overall, we had an estimated 38,000 cases of leptospirosis in the Caribbean region, with an average of 1,400, 1,500 cases yearly in the region, with islands like Cuba, Dominican Republic, Haiti, that has the largest population, also having the largest number of cases, which changes when we look in incidence, not only number of cases. And then if people are not super familiar, but in the panel B, we have the Lesser Antilles that are also tiny islands with very high incidence compared to the larger countries. We also identify that leptospirosis was associated with environmental factors. For example, biodiversity loss was the most important factor in this model, significantly associated with leptospirosis. Rainfall or extreme weather events like floods, water-related, were positively associated but not significant in this. So showing that although we could identify, we could predict, there are still some pitfalls when we're using surveillance data. And usually those will follow because we're using notification, so routine-based surveillance. So those cases were, we might miss mild or asymptomatic cases, or we have problems with laboratory that was not being able to capture or have the capacity to run all the tests that they need. So with all of that, we know that this is it gives us some estimates, but we still need a lot of caution when we're interpreting these results. And that led us to think, okay, so what we'll do now that we have access to serological survey. And that's focusing on the Dominican Republic. It's also a country in the Caribbean. And we're gonna actually focus even more in those two provinces that are highlighted, so Espaillat and San Pedro de Macorís. And we had a survey that was conducted between June and October 2021. And we collected blood samples and questionnaire data, combined these again with similar variables that were used in the other models, and using spatial methods, try to identify patterns of this epidemiology. What changed now is that we're not focusing only on the cases that we know, but we have a whole population that we know which ones are positive, which ones are negative. And as this was done in a design that is population representative, we can better understand some epidemiological behaviors of this disease. And first step that we did with this data was trying to understand what was the spatial structure. So the difference here is that I want to see if things that are closer together are more similar than things that are further apart. And for that, I use a clustering analysis and identify possible hotspots or clusters of seropositivity of leptospirosis. And after that, once we identified where were those clusters, we compared the population living inside the clusters and outside the clusters to see if there was difference in distributions of groups among these two populations. So cluster analysis, and that. Yes, I identified some clusters using these methods. This was a flex scan. Those are the areas highlighted in the yellow circle. And those areas have a higher risk and a significant higher risk of finding a positive individual compared to the other areas. And running a logistic regression to compare populations inside and outside the clusters, we identify several things that were associated with this. I'm not going to make you understand all this table because it is a large one, but the most important things is that gave us an idea that the population inside the cluster were more likely to be poor or resource limited, living in remote areas and associated with rural environment, which is a little bit surprising compared to what we initially thought, but it's what we found. So we identified clustering and we identified characteristics of this. So we have the idea that this is a data set that has some spatial structure and that allowed us to move on to a spatial model. First spatial model that we tried was to identify variation in the association of leptospirosis seropositive with the drivers of the disease. And we used for that a geographically weighted regression. So variable selections were informed by a non-spatial model, just a simple mixed effects model. And then we did the geographically weighted regression. What we can see in this panel is by using the geographic weighted regression, so allowing the odds ratio to vary across space, I can see that the drivers of the disease don't impact seropositivity in the same way. So they will vary, and even inside, for example, group age, that is A, B, C, and D, you have a different trend across space, with some going north to south, others going west to east. And the risk for each individual in this location will be a combination of all of this variation at the same time. And that led us to the last study using this data. That is this one. So the idea was, okay, I know that the odds ratio varies across space, but can I use that to predict seroprevalence? And how can I do it in the best way? So before jumping into the model itself, I tested a few things that would be interesting to check. So if I used only this variable selection that I used for initial geographically weighted regression, I would be doing model A. That is not necessarily spatially informed because I use the non-spatial methods to select my variables. So that's what we have here. But then I tested, because of the clustering analysis, also informed us in a different set of variables, and that was spatially informed. So we tested if combining those variables to the initial model, if that would impact and have a different result. So this is spatially informed. And then finally incorporating survey questionnaires to create a risk profile based on that. So results from model A and B, so non-spatially informed model A and spatially informed model B, are quite similar, but they're able to predict a different distribution of the disease with a lot more information, because now it's at the subnational level, and we can see how it varies with the capitals, the more close in a spila, so in the top, around that blue area in the center, that's the capital, we have a lower risk, but as we go to more remote areas, we have a high risk. We change this a little bit in San Pedro de Macorís, that the capital is in the bottom where we have this greenish area, and all in the model B, we have further remote areas increasing risk, but not the same in model A. So having difference when we spatially informed our model. If you're wondering why this is not a continuous map, is because the only populated areas are showed. So I excluded areas that were not populated. That's why there's a little bit patchy. And finally, when incorporated the questionnaire variables, we created those charts where I have the risk that can be assessed. So seroprevalence risk assessed based on specific characteristics. So I know that freshwater exposure was important for spila. So if there is or there isn't freshwater exposure, and the age, and it's female or male, and I can actually make direct comparisons between the groups, because I can see the highest group, that is freshwater exposure, male, elderly, with the lowest, that would be negative red exposure, female, young population. This is very useful considering in a clinical setting, because if I have someone that comes with symptoms that are similar to leptospirosis, I can assess the risk of the disease based on those charts. So it would be very interesting to use. So overall, the sero surveys, especially combined with spatial data, we're able to have a more comprehensive characterization of the epidemiology in country and could give us a better understanding of the subnational distribution as well, which is very useful when you're thinking in resource allocation, because I know now areas that should be prioritized. Help identify hotspots, spatial variation, and also to predict risk. And modeling S was crucial to reveal those nuanced patterns. I think that was all. I would like to thank all my supervisory team that helped me through all those studies, and also collaborators and partners in country that helped us collect the data and analyze it and make a sense of all of these. Thank you.